Hello, my name is Julia Sloan, and this summer I did a surf with the Moscato Group at the University of Newcastle in Newcastle, Australia. So today I'm going to be talking to you about increasing efficiency of memetic algorithms. So here's a little roadmap of what we're going to talk about today. So first I'll get into some background about what are memetic algorithms, and also I'll explain what continued fractions are and how they relate to our algorithm. Then I'll talk about data sets and why they're important in machine learning, and I'll talk about two data sets that I've looked at in particular. And then I'll have a little conclusion where I summarize everything I've gone over and talk about future work and say my acknowledgements. So first, what are memetic algorithms? So they're a machine learning tool that were developed by my mentor, Professor Pablo Moscato, at Caltech in the 1980s. Like many other machine learning tools, they're used to approximate and predict functions. So pretty much anything you can think of can be modeled by a function, so this has a wide variety of applications. So everything from US election results to drug distribution in the body to sommelier rankings of wines. So really anything you can think of, you can use this method for. So how does it work? How do memetic algorithms work? So the term memetic, you can think of as analogous to genetic. So whereas the word genetic comes from genes, which we know from evolution, they're passed down from generation to generation and carry information. What we call memes are really similar, except they carry more information. But you can think of them as essentially the same thing. So I have a little flowchart here of how the algorithm works on a basic level. So first you generate your initial population, and this is kind of similar to the ideas behind evolution, where everything is constantly evolving and improving and generating more solutions. So the initial population contains potential solutions to whatever you're trying to approximate. So these solutions are functions that will approximate your goal function. So you generate the initial population, evaluate the population using whatever metric you want. We typically use mean squared error, or MSE. You mutate the solutions, so change them a little bit. And then you ask, have we reached the maximum number of generations? Typically, we use 200 generations because that's sufficient to give us good results. If you're there, you output your best solution so far. If not, you select some of your best solutions, mutate them a little bit further to get a new population. So this is like your new generation, and you continue the cycle. So in this way, it behaves a lot like evolution, and it continues to improve from each generation to the next, giving us better and better results. So now I'm going to talk about continued fractions a little bit. So continued fractions are similar to normal fractions, but with normal fractions where you would have something like 1/2, 1/2. With continued fractions, your denominator is another fraction. So it continues to expand like this and continue down the line. So I have two examples of continued fractions here, and you can see that they kind of just continue on rather than stopping. These can be finite or infinite, and what we refer to as the depth of a continued fraction is how many kind of sub-fractions are in it. So if we look at this one on the right with b's and a's, if we just had b naught, it would be depth 0 because there's no subfraction there. If we had b naught plus b1 over a1, that would be depth 1 because there's one fraction there, and so on. So increasing the depth of the fraction means your continued fraction is growing, which is helpful because it allows you to more accurately represent functions. So how is this helpful to us? Well, Euler told us that you can represent the sum of products as a continued fraction. So the sum of products is a polynomial, which is a class of functions, but only a type of functions. It's not all functions. And we really want to be able to replicate any kind of function we're going to come across, whether it's a polynomial or a trigonometric function or anything like that. So the way that we use this kind of idea is instead of using just a0, a1, and a2 as constants, we actually substitute in other expressions of x. So by doing this, instead of just modeling a sum of products, we can model whatever kind of function we come across. So the reason that I'm bringing this up is this is kind of the internal representation of functions that our algorithm uses, which is useful because it allows you to store different types of information without having to explicitly state what is going on. And because we use continued fractions, we refer to our algorithm as continued fraction regression. Okay, so now that we have that background in our pockets, we're going to move on to data sets. So a data set is basically a collection of a large amount of information. It can be anything you want. The examples I brought up before about the election and wine and those things, those are all data sets. So there are two main aspects when you're designing an algorithm. There's the accuracy, so how well it approximates the function you're trying to approximate, in this case, and the efficiency, so how much time and space it takes up as it does so. Now, our current continued fraction regression algorithm is very accurate, but it's not as efficient as it could be. Sometimes it takes hours to run it on a single data set. So by running our algorithm, on a variety of different diverse data sets, we can see where it performs well and where it performs poorly. And by performing well, I mean it's both accurate and time efficient. So as part of my project, two of the data sets I looked at were proteins and plates. And I'm going to get into those more as we go on. First, I'm going to talk about the proteins data set. So a little bit of background here. On the right, I have a diagram about protein folding. So this chain on the left, where it says unfolded, is an amino acid chain, which makes up all proteins. And on the right, we have a final folded protein. So each amino acid chain starts as this long string, and through some complicated series of events, forms a specifically structured folded protein in a 3D structure. So this data set that we're looking at has 52 different proteins, where the protein is an amino acid chain, and 1,250 potential 3D structures for each of those chains. It also gives us the energy associated with each of those final structures. And our goal here is to take that amino acid chain that we're given and predict which of those many potential structures it will actually end up taking on. So here we have the results of running our algorithm with this data set. What you see in orange is what our algorithm predicted, and what you see in blue is the actual function that we're trying to approximate. So on the left here, we have our prediction at depth 0, and you can see that the function we're trying to approximate has this shape where it goes up kind of continuously, and our approximation is just a flat line. So this is not a great approximation. However, this is only at depth 0. So by increasing the depth of the continued fraction, we can get a slightly better estimation. So on the right here, we have the same result on the same protein at a higher depth. So you can see that it does a slightly better job, especially on the left side, which are lower energy potential structures, but it's still not great. So this is on the first of 52 proteins, but I also ran experiments with the second of 52 proteins. So this protein is called 1AF7. And on the left, again, we have the depth 0 result. And you can see, again, it's just a flat line that doesn't really approximate the function very well. And on the right, we have the depth 2 result, which is not exactly a flat line, but still doesn't really approximate the function well. So these results were a little bit disappointing. We know that our algorithm can do better than this, but we did do a final experiment with the proteins data sets. So we wanted to know if we could use the function we predicted from one protein to model the energies of another protein. So the graph we have here in orange, we have the prediction we created for the 1ABV, the first protein, and we have the energy of the second protein. And you can see they're kind of nowhere near each other. So this didn't really pan out. It could be for a number of reasons. As we saw before, our approximations weren't really that great for the same protein itself that we were trying to model. So it's a little bit bold to hope that it would work for other proteins as well. But a potential explanation is that some proteins have shared characteristics with each other. So maybe we need to split the proteins up into 3 to 5 groups and kind of model each group individually with its own energy function. That's one potential avenue that we could take with this. But as of right now, we're not getting great results. So we'll move on to our plays dataset. So the plays dataset contains 185 plays from the Shakespeare era and 16,384 words. And it also gives us the percentage each word is of all words in the plays. So basically the frequency that each word appears for each play. So the goal here is to take which words most commonly appear in each play and determine what year that play was first performed. Okay, so on this slide, we're going to go from the left to the right. So run 1, we ran our algorithm on the data set using all of the plays and all of the words, so lots and lots of words, at depth 0. And you can see it does a pretty good job of following the trend. The MSE is relatively low at 32.37, but it did take 2 hours, which is kind of a long time. So to move forward with these results, I decided to take the words that were used in the function producing these orange dots. So the words used in our actual model, and only use those words as an input to our algorithm rather than using all words. So in run 2, you can see it's titled CFR algorithm using words of first result. So here we're using many, many fewer words, only about 7, I think. And you can see it's a much better fit. The MSE is down to 23.32, and this only took a minute. So the time efficiency is very much improved. And again, this is at depth 0. So I wanted to test my luck, and I tried this again. So this time I used the words that were used in the function produced by the second run, and I fed those into the algorithm to get the third run. So in this case, the MSE did go up slightly, but it's essentially the same result as we had in run 2. So I decided to stop there. And again, this only took 1 minute, which is really good. And these are all at depth 0. So you can imagine that if we increase the depth even further, we might get even better results. Another experiment I did with this data set is I ran it with our algorithm using the 50 most common words across all plays. So it's also worth noting that for these experiments, I used a restricted date range from 1585 to 1610. And the reason I did this is because we saw that most of the plays were concentrated within this date range, and we wanted to exclude any potential outliers, which we figured was fine because most of the plays were first performed within this range anyways. So on the left here, we have the result at depth 0, and you can see it does pretty well. It certainly follows the trend. There are a lot of dots that are directly on the blue line. And on the right, we have the result at depth 0. So it's a little bit better. We went from an MSE of 23.87 to an MSE of 20.87. So slight improvement there. And especially compared to the results we got in the proteins dataset, we're doing very well here with this dataset. So in conclusion, our results with the proteins dataset were not great. This could be due to a flaw of our algorithm or some kind of nuance with the dataset that we're not picking up on, like perhaps the subgroups that I mentioned before. To kind of test what's going on in the future, we could test our algorithm with similar datasets, maybe in size or other molecular biology datasets, to see if it's an issue with our algorithm or just a particular anomaly with this dataset. And in terms of the plays dataset, our current algorithm does quite well at predicting the play production years. One thing we do want to do in the future is examine any obvious outliers we can see and see if they have anything in common. So for example, if they were written by the same author, maybe he has a particular way of writing that was just unique from other authors at the time, or maybe they're from the same city, which had its own style, also unique at the time, which could both contribute to making those points be outliers. And finally, I want to say a couple of words of thank you. So to the Moscato group for having me, letting me join their lab this summer. Professor Pablo Moscato and Mohammed Haig for teaching me about the algorithm and how it works and all of these machine learning concepts. My fellow SURF students, John Corrales and Kevin Huang, and Professor Hugh Craig, who is a linguistics professor at the University of Newcastle, working with us on the Plays dataset. And also the SURF office for putting this whole thing together, as well as Samuel P. and Francis Crown for sponsoring my SURF. Thank you.